Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination question paper for the subject Animal Biology and Biotechnology conducted in the year 2021. I will be discussing only the biology part. If you are new to my channel, please do like, share and subscribe. So coming to the first question. Open vascular system is usually found in. So open vascular system is primarily found in invertebrates. Here the blood flows freely through cavities since there are no vessels to conduct the blood. So it is seen in the case of crabs. Answer is option A. Then in thermal stratification, the middle region which shows vertical temperature change is called. So the thermal stratification of lakes refers to change in temperature at different depth in the lake. Now metalimnion or thermocline, where temperature changes with depth. So it is answer is option C, that is metalimnion. Then in bilateria development, splitting of solid mass of mesodermal cells to form the body cavity result in schizocylone. Answer is option A. Then, with reference to T cell receptor, which of the following statements are true? So it is required for helper T cell to stimulate B cells in humoral response. So that is correct. It is required for killer T cells to recognize antigen. That is also correct. It recognizes antigen fragments and MHC molecules independently. So T cell recognize an antigen when they bound with major histocompatibility complex molecules on the surface of other cells. So option, that is the statement 3 is not correct. It has a domain organization that is roughly similar to that of immunoglobulin domain. That is also correct. So 1, 2 and 4 are correct. So the option is option C. The next one, acridine orange is used to label. So acridine orange is cell permeable, which allows the dye to interact with the DNA by intercalation or RNA via electrostatic attraction. So both DNA and RNA is the correct answer. Then nude mice used for developing tumor models lack one of the following glands. So nude mice, it lacks the gland thymus. Then the macrophage-rich mass found at the site of injection of an adjuvant is called. It is called as granuloma. And what is myeloma is cancer that forms in a type of WBC called plasma cell. And hybridoma, it is a culture of hybrid cell that result in the fusion of B cells and myeloma cells. Then next one. Hyaluronic acid is a multimer consisting of alternative residues of. So it consists of alternative residues of D-glucuronic acid and N-acetylglucosamine. Answer is option B. Then which of the following statements hold true for placenta in mammals? So option belongs to fetus as well as mother from the respective ends. So that is the case of placenta in mammals. And next one, in a DNA fragment, the following sequence is converted to this. This is called as. So here, if you see this cysteine, is converted to guanine here. Is converted to guanine. So here a cysteine is a pyrimidine and guanine is a purine. So a pyrimidine is converted to purine, so it is the case of transversion. Answer is B. Vesicles transporting proteins from plasma membrane to endosomes will be coated by. So it will be coated by clathrin. So COP1 is a transport protein from the cis end of Golgi complex back to rough endoplasmic reticulum where they were originally synthesized. And COP2 is a type of vesicle coat protein that transport proteins from the rough endoplasmic reticulum to the Golgi apparatus. Then next one. Ora serrata is related to which organ? So ora serrata, it is a junction between the choroid and the ciliary body. It is found in the case of eyes. Answer is option. Then which of the following is true about blood transfusion reaction, erythroblastosis pigtalis and autoimmune hemolytic anemia? They are all type 2 hypersensitive reactions. So answer is option A. Then which one of the terms describes a mitotic cell division giving rise to two daughter cells without increasing the size of the original parental cell? So it is cellulation. What is hyperplasia? Hyperplasia is an enlargement of an organ or tissue caused by an increase in the amount of organic tissue that result from cell proliferation. Blastocele is fluid-filled cavity that forms in the blastula. And cytokinesis is part of cell division process during which the cytoplasm of a single eukaryotic cell divides into two daughter cells. Then anti-aging pills like TA65 primarily activate. So they primarily activate telomerase enzyme. So a telomere, that is a region of repetitive sequence at the end of chromosomes. And telomeres, they protect the end of the chromosome from DNA damage or form fusion with neighboring chromosome. And with every cell division, telomeres shorten. This blocks further cell division, induces senescence, apoptosis, oncogenic transformation of somatic cells, and affecting the health and lifespan of the individual. So an enzyme called as telomerase is responsible for maintaining the length of the telomeres, and it maintains the length of the telomeres by addition of guanine-rich repetitive sequences. So it is anti-aging pill TA65 primarily activate telomerase enzyme. Then the 9+2 pattern found in cilia or flagella occurs in every animal phylum except. So the 9+2 arrangement, it refers to the 9 fused pair of microtubules on the outside of the cylinder and the 2 unfused microtubules in the center. So 9+2 found in cilia or flagella occur in every animal except arthropoda. It is not seen in the case of arthropoda. Then in Drosophila, the sex is determined by. So this is a usual question. Either they ask it directly as what has been asked here or in a different way. So the ratio of the number of X chromosome to the sets of autosomes. Answer is option C. Then which of the following method is better suited to identify a putative enhancer elements? So if you see for chromatin immunoprecipitation or CHIP, it is an antibody-based technology used to selectively enrich specific DNA binding proteins along with their targets. So here for identifying putative enhancer elements, you can use the CHIP P300 antibody technology. And CHIP, it is used to investigate particular protein-DNA interactions. Then EMSA, electrophoretic mobility shift assay, that is also used for determining if a protein or a mixture of protein is capable of binding to DNA or RNA sequence and can indicate more than one protein molecule is involved in binding the complex. So why we choose CHIP instead of EMSA method? So CHIP is now considered the gold standard measure of DNA binding because it captures protein DNA interaction that occur in the intact live cells. Whereas EMSA, it does not provide information about the sequence specifically are occupied, which sequences, that is, it does not provide information about which sequences that are specifically occupied by the protein of interest. And EMSA, it is completely an in vitro technique. Whereas CHIP, it is in live intact cells. So the correct option here is option B, that is CHIP P300 antibody. Then if a plant with genotype A, B, capital A, small a, capital B, small b is self-fertilized, the probability of getting capital A, capital A, capital B genotype will be A and B are not linked. So this is the genotype of the parents. So this is self. So you cross it with the same parent of the same genotype. And these are the gametes over here, and this is the Punnett square showing the likely possibilities. So you will get capital A, capital B only one time in the Punnett square. So out of these 16, one will be capital A, capital B. So the answer here is option D. That is 1 out of 16. Then plants adapted to swampy environment meet oxygen requirements of their roots by means of a specialized tissue called. So it is the tissue called as the parenchyma. Parenchyma is a spongy tissue that forms spaces or air channels in the leaves, stem and roots of halophytic plants. The presence of parenchymatic tissue help in exchange of gases between the shoot and the root system. The next one, consider the phylogenetic relation among the following species. Which of the following is most closely related? So this is the phylogenetic tree, and phylogenetic tree, it is a diagram that represent evolutionary relationships among these organisms. And phylogenetic tree are just hypotheses and they are not any definite facts. So a phylogenetic tree, you have got the root. This is called as the root of the phylogenetic tree, and it is the most common ancestor of this one. This one is the most common ancestor of all these animals. That is the branch point. This is the branch point. Okay. So that particular branch point, it represents the common ancestor of all these animals. So this is the ancestor and these are the present day plants. I forgot to tell that in the beginning. This is the ancestor and these are the present day animals. Sorry. And this is the branch point and these are the branches. Okay, these are the branches, this is the branch point. So here this point, that is this branch point, it represents the most recent common ancestor of these two animals. This branch point, it represents the most common recent ancestor of these two animals. So these are the branches, this is the branch point, this is the root, and it represents ancestors. These are the ancestors and these are the present day animal species. Now each branch point, that is here, it represents a divergence or splitting apart of a single group into two descendant groups. So this branch point, it shows a divergence, that is this that these two animals originated from this particular ancestor. So that is what is shown here. And at each branch point lies the most common, that is recent common ancestor of all the groups descended from that branch point. Now, two species are more related. Two species are more related if they have a more recent common ancestor and less related if they have a less recent common ancestor. Now, if you take these two species, they are more related. Why? Because they have a more recent common ancestor. But these two species, they have different common ancestor. That is white-tailed deer. That is having a different common ancestor. That is, it passes this particular ancestor and then only comes over to this ancestor. But blue whale also, it crosses this particular ancestor and then comes to this particular ancestor. So white-tailed deer and blue whale, they are not more related. Okay, they are not more related because they have a less recent common ancestor. So this is their ancestor and these are the ancestor of blue whale and white-tailed deer, and they have a less recent common ancestor. So these two are not related. So what you have to do is follow the lines of both the pair of species backward in a tree. That is, these two species, if you follow the line, you will reach a common ancestor over here. For white-tailed deer also, you will reach a common ancestor over here. But if you take white-tailed deer, you will reach the most common ancestor here. And blue whale also the most, if you trace backward, you will reach the most common ancestor over here. That is white-tailed deer and blue whale, it converges after its junction point. Okay, this is the junction point. So it converges only after the junction point. So we can say that Angus cattle and white-tailed deer are more commonly related than white-tailed deer and blue whale. So now coming to the options. So first one is white-tailed deer and blue whale. I told you already, white-tailed deer and blue whale, they are not closely related. Then you have pygmy hippopotamus and wild boar. Here, this is pygmy hippopotamus and this is wild boar. That is also not commonly related. Closely related. Then you have white-tailed deer and Angus cattle. White-tailed deer and Angus cattle, they both have a common ancestor. If you trace back this particular line, they will reach a common ancestor. So that they are more closely related. Whereas wild boar and alpaca, they are also not closely related. So the answer here is white-tailed deer and Angus cattle. So the answer is option C. The next one. The genome of Middle East Respiratory Syndrome, COV, MERS-CoV is A. So it is an RNA with positive strand. So answer is option A. Then identify the mismatched pair. Tundra permafrost, prairie epiphytes, savanna acacia trees, coniferous trees, evergreen trees. So prairies, they are grasslands and epiphytes and ephemerals are found in the desert. So the mismatch is option B. Then if polyuracil RNA is used in vitro translation reaction, the generated polypeptide contains. So if polyuracil, that is poly-U, RNA is used in vitro translation reaction, the polypeptide contains, because UUU codes for phenylalanine, so it will naturally contain the polypeptide with phenylalanine. Then the biogeochemical cycle of phosphorus differ from the cycle of carbon and nitrogen in that. So the nitrogen cycle, it is a biogeochemical cycle by which nitrogen is converted into multiple chemical forms as it circulates among stratosphere, terrestrial and marine ecosystem. Whereas the carbon cycle, it circulates between the biosphere, pedosphere, geosphere, hydrosphere and atmosphere of the earth. Whereas phosphorus cycle, it cycles through biosphere, hydrosphere, lithosphere but no atmosphere. Whereas the nitrogen cycle and the carbon cycle has got atmosphere in them. Phosphorus cycle does not have atmosphere. So the answer is option A, that is phosphorus cycles through biosphere, hydrosphere, lithosphere but not atmosphere. Then which among the following is not a professional antigen presenting cell? So professional antigen presenting cells are immune cells that specialize in presenting an antigen to a T cell. The main professional antigen presenting cells are dendritic cells, macrophages and B cells. So the one which is not a professional antigen presenting cell is fibroblast cells. Then the heaviest naturally occurring element is, it is uranium. Answer is option B. Then water walking occurs in many insect groups except, so it occurs in many insect groups except Hymenoptera. Then which one of the following is the correct food chain? So it is phytoplankton, zooplankton and then fish. Phytoplankton being eaten by zooplankton and zooplankton in turn being eaten by the fish. So answer is option A. Then in an immunization experiment, there are 4 different groups of mice. Group 1 mice are administered with keyhole limpet hemocyanin. Group 2 mice received azobenzene arsenate. Group 3 mice received lipopolysaccharide and group 4 mice received dextran. After 4 weeks, sera were collected and tested for antigen-specific IgG1 and IgG2A antibodies by ELISA. Which of the following groups will show the maximum titer? Now before coming to the answer, let me tell you antibody titer. It is a type of blood test that determine the presence and level, that is this level, that is titer of the antibodies in the blood. This test is carried out to investigate if there is an immune reaction triggered by antigens in the body. Now the answer is keyhole limpet hemocyanin primed. That will show the maximum titer. Why? Because it is a protein and protein is a powerful antigen than other groups. So it will give maximum titer. So the answer is option A. Then next one. Which of the following among alkali metal is most reactive? So the reactivity of group 1 elements. All these are mainly group 1, sodium, potassium, cesium are all group 1 elements. So the reactivity of group 1 elements increases as you go down the group. Why? Because the atoms become larger, the outer electron becomes further away from the nucleus and the force of attraction between nucleus and the outer electron decreases. The outer electron is also lost easily. So the reactivity increases down a group and cesium will be the most reactive. Then relationship of an epiphyte with its host is an example of. So epiphyte depends on their host for structural support but does not derive nourishment from them or harm them in any way. So it is the case of commensalism. Answer is option C. That is, one species is benefited but the other species is neither harmed nor benefited. Then which of the following is not an example of selective media? So trypticase soy agar is not an example of selective media. It is a general purpose, non-selective, non-differential medium that support growth of all microorganisms that do not require special nutrients. So next one. The major difference between food web and food chain is. So food chain is a single linear pathway and food web is an interconnected pathway. Answer is option C. Then in E. coli, which of the following sigma factor is involved in the transcription of the housekeeping gene? So it is sigma 70. Answer is option A. Then find the weight of H2SO4 in 1200 ml of a solution of 0.4 normal strength. So you are given the volume, that is 1200. You have to find the mass. So number of hydrogen ions in H2SO4 is 2 and equivalent mass is equal to molar mass of the solute divided by N. And equivalent mass is equal to 2 plus 32 plus 64 divided by 2, you will get 49 grams. And normality is equal to mass of the solute into 1000 divided by equivalent mass of solute into volume in ml. So normality, if you calculate, you will get 23.52. So the answer here is option A, that is 23.52 grams. Then coming to the next question. Cryptochromes are fluorophores of plants that are sensitive to. So they are sensitive to blue light. Answer is option C, blue light. Then which of the following is not true with reference to limbic system related memory responses in vertebrates? So human beings with hippocampal damage by neurodegeneration retains good short-term memory but poor long-term memory. So that is the answer. Answer is option C. Then which of the following compounds will be optically active? So that there is an asymmetric chiral carbon atom. If you see lactic acid over here, there is an asymmetric chiral carbon atom. Okay, so that will be optically active. This one, it has got 4 groups attached to the carbon atom. 4 different groups are attached to the carbon atom. Whereas if you see mesotartaric acid, succinic acid and chloroacetic acid, it is not the case. You can see 2H over here in case of chloroacetic acid. So the answer is, the compound which will be optically active is lactic acid. Answer is option C. Then production of ammonia by microbes is tested by. So it is tested by Nessler's test. Answer is B. Then phosphoric acid has 3 pKa values. The number of pKa values that a phosphate group can have in DNA or RNA is. So the answer is 1. Option B, that is 1. Then twin studies in humans are useful because they allow genetic as opposed to environmental influences on the variation in a trait to be estimated. Answer is option D. Waterlogging is a phenomenon that refers to saturation of soil root zone. Answer is option C. Then which is the correct sequence of arrangement of type of WBC in decreasing order in terms of number per millimeter cube of healthy human blood? So the answer is neutrophil, then eosinophil, and then basophil. Neutrophil, it accounts to be 2500 per 8000 to 8000 per mm cube. Eosinophil, it is 500 mm cube, and basophil, it is 25 to 100 mm cube. Then gray crescent is present in. So gray crescent, it is a structure formed on the opposite side of sperm entry in a frog egg. So it is present in zygote of frog. Answer is 9. Option A. Then next one. The atoms of the element having the same mass number but different atomic number are called. They are called as isobars. Answer is option B. Then the DNA polymerase with 3' 5' exonuclease activity is DNA polymerase 1. So answer is option A. DNA polymerase 1. Then you have a mixture of proteins with the following properties. So protein 1 is 58 kDa, 2 is 28 kDa, 3 is 13 kDa, and 4 is 10 kDa. Protein 3 and protein 4 exist as a heterodimer. So these, that is 3 and 4, they exist as a heterodimer, while protein 1 and 2 exist as monomers in the solution. What is the order of elution of these proteins on a gel exclusion chromatography? So a gel exclusion chromatography, it separates proteins solely on the basis of molecular size. Larger protein molecules emerge first from the column. Then before that, what is a homodimer? It comprising of the same polypeptide, and heterodimer, it is comprising of two different polypeptides. Now it is given that protein 3 and 4 exist as heterodimer and protein 1 and 2 exist as monomers. So in gel exclusion chromatography, separation takes place based on size only. The protein will always separate on the basis of its native size. There is no denaturation of the proteins here. And it will not get separated into its polypeptides while passing through the column. Since protein 3 and 4, it exists as heterodimers, that is these two. 3 and 4, they exist as heterodimer. While passing through the column, the dimers doesn't split up. They will elute in their native form, that is 28 kDa protein. So that is 17 plus 10 kDa, you will get it as 28 kDa protein. Now 28 kDa is also the molecular weight of protein 2, that is over here. This is also 28 kDa. So protein 2, 3 and 4 will co-elute as a single peak, and you will get two peaks on the chromatogram corresponding to protein 1 and co-eluted protein 2, 3 and 4. So the answer here is option B. That is protein 1 will elute first, then because that is of the larger molecular weight, that is 58 kDa when compared to 28 kDa, which is lesser molecular weight than protein 1. And then protein 2, 3, 4 co-elute as a single peak. The chromatogram will depict two major peaks. Answer is option B. Then next one. Which of the following can precipitate antigens? So FAB2 can precipitate antigen. Monovalent FAB fragment have one antigen binding site, whereas divalent FAB fragment have two antigen binding site that are linked by disulfide bond. So this is a repeated question. A similar question was asked, I don't know which year, but it was asked, I think in integrated MSc PhD question paper. Okay. Transamination of which of the following amino acids give rise directly to one of the intermediates of the tricarboxylic acid cycle? So it is glutamate. Answer is option C. Then pap smear test is used for screening of cervical cancer. Answer is option C. Then an example of a compound that acts as an antioxidant in packed food is, so it is butylated hydroxytoluene or BHT. Then for enzymes obeying Michaelis-Menten relationship, a plot generated using 1 by V0 versus 1 by concentration of S is known as, it is known as Lineweaver-Burk plot. Answer is A. Then which of the following compounds will react with ammoniacal silver nitrate? So it is 1-butene. So 1-butene, it being a terminal alkyne, it reacts with ammoniacal silver nitrate solution to produce a white precipitate, that is 2-butene. And 2-butene, it is a non-terminal alkyne, so it does not react with ammoniacal silver nitrate solution. Then next one. For a given DNA sequence, this is also a repeated question. I don't know in which question paper, but it was asked in the same university examination. I don't know which question paper, but this is also a repeated question. So, for a given DNA sequence, they have given a DNA sequence. Which one of the following serve as forward and reverse primer during PCR amplification? So, they have given the sequence in 5' 3' direction. So, 5' 3' direction. You have got the complementary sequence in 3', 5' direction. So here, because the DNA polymerase, it will add nucleotides to the 3' end. Forward primer is over here. That is attached to the 3', 5' region of the, that is attached to the complementary strand of the given strand. That is in the 3', 5' direction. So it will be 5' to 3' direction that this particular forward primer will be synthesized, 5' to 3' direction. So the 5', 3' direction, if it has to be synthesized, it should be complementary to 3', 5' strand. So the primer has to be in 5', 3' orientation, and it should be complementary to the 3', 5' strand. So we can see that the primer is the same as the sequence of the 5', 3' strand of the DNA. So the forward primer remains the same as that of the given strand. So GGAACCTT and GGAACCTT. So this particular primer sequence is same as the sequence of the strand in the 5', 3' direction. So that is the forward primer. Whereas the reverse primer, it synthesizes from this direction. That is, here is the 3' end and here is the 5' end. Then the reverse primer, which is complementary to this particular strand, that is 5' to 3' strand, will synthesize, will amplify the DNA strand from here, that is 5' to 3'. So it is the reverse complement of this particular strand, that is 5' to 3' strand. So this is the sequence of the reverse primer, which is complementary to this particular strand. Okay, so GATC, that is the reverse primer read in the 5' to 3' direction. So answer is option D. GAACCTT and reverse primer is GATC. Then next one. In the absence of any gonads in a fetus, the external genitalia will be female. Answer is option B. Then at pre-B cell stage immunoglobulin, heavy chain polypeptide are expressed on the cell surface along with lambda 5 and VP or VPRE-B. That is in the case of B cell development. Then match the following. Teloleucithal egg. So it is of bird. Answer is 1, 3. Then next one, isolecithal egg is for sea urchin, centrolecithal egg is for insect, and mesolecithal egg is for frog. So the answer is option B, 1, 3, 2, 1, 3, 4 and 4, 2. Then which one of the following technique is used to determine the size of a native protein? So it is gel filtration. Answer is option C. Then Bell's-Wiley sperm sexing technology is developed based on. So Bell's-Wiley sperm sexing technology, it is an effective means of producing progeny of predetermined sex in cattle, sheep and laboratory animals. The method is based on flow cytometric separation of X and Y chromosome bearing sperms and subsequently be used for surgical, intratubule or intrauterine insemination. So here the answer is, it is based on flow cytometric method. So answer is option B. Then next one, a cross between two inbred lines, one with dark blue flowers and one with bright white flowers, produce F1 offsprings that are light blue. What is when the F1 progeny are self, a 1:2:1 ratio of dark blue to light blue to white flowers is observed. What genetic phenomenon is consistent with these results? So this is the parent genotype, that is capital B and small b small b. This is the F1, that is capital B small b. When you self the F1, you will get 1:2:1, that is 1 dark blue, 2 light blue and 1 white flowers will be obtained, and this phenomenon, it is called as incomplete dominance. Answer is option D. So the phenomenon in which the true breeding plants crossed to produce an intermediate offspring is known as incomplete dominance. Here capital B small b is of intermediate phenotype. So next question. A substance that diffuses through the egg and by virtue of its concentration determines the developmental fate of the embryo is known as. So it is known as morphogen. Answer is option D. Then Cas9 is. So it is an RNA guided endonuclease that can be used for site specific cleavage of double stranded DNA. It is not a restriction enzyme. The guide RNA is designed to find and bind to specific sequence in the DNA. So the answer here is option D, that is an enzyme that cuts DNA at a specified site through a guide RNA molecule. Answer is option D. Then next one. Which one of the following statements about mitochondrial protein import is true? So the mitochondrial targeting sequence are always present on carboxy terminus. Not carboxy terminus, it is on the amino terminus. So that is not correct. Then mitochondrial protein cross the membrane in an unfolded state. So B is correct. Then all of the mitochondrial proteins are synthesized by mitochondria itself. Although mitochondria do synthesize a few of their own proteins, the majority of the proteins they require are encoded in the nuclear genome. Then chaperones are not required for mitochondrial protein import. Chaperones, they facilitate pre-sequence translocation, unfolding of the cytosol or localized domains of precursor proteins, then movement across the mitochondrial membrane, and finally folding of the newly imported proteins within the matrix. So the answer is option B. Then, with reference to the 2018 Nobel Prize in Physiology or Medicine given to the immunologists James Allison and Saku Honjo for their discovery of cancer therapy, which of the following statement is true? So the answer is option C, that is CTLA4 and PD1 protein function as brake on the immune system. The next one, which of the following is not a potential pre-zygotic reproductive barrier? So it is hybrid infertility. It is a post-zygotic mechanism. Answer is option C. Then which of the following statements about biological membranes is not true? They contain carbohydrates covalently bonded to protein and lipid. Then they are very large sheet-like structures with closed boundaries. They contain symmetrical lipid bilayer. So it's not true. So answer is option C, that is not true. It is asymmetric lipid bilayer with different membrane lipid composition in each monolayer. So answer is option C. Then which one of the following is a DNA vaccine? So HPV is a DNA vaccine as well as hepatitis B is also a DNA vaccine. So both C and D are DNA vaccines. Then which one of the following is a marker for early endosomes? Now early endosomes, these are organelles that receive macromolecules and solutes from the extracellular environment. The major function of early endosome is to sort these cargoes into recycling and degradative compartments of the cell. Then each stage of the endosperm maturation is marked by a unique set of proteins. Some members of the RAS superfamily, that is specifically RAB5, RAB7 and RAB11 are markers of the early, late and recycling endosomes. So RAB5 is for early, RAB7 is for late and RAB11 is for recycling endosomes. So the answer is early endosome, it is RAB5. Answer is option A. Then germ cell aplasia is best described by which one of the following? So answer here is option A, that is infertility without sexual abnormality, normal level of LH and testosterone, low level of inhibin B, elevated levels of FSH and azoospermia. So Sertoli cell-Olli syndrome, also known as Del Castello syndrome or germ cell aplasia, it typically occurs in males between the age of 20 and 40. In this syndrome, only Sertoli cell lines the seminiferous tubules of the testis and the patient have very low or absent spermatogenesis. So the patients are typically normal on physical examination as this condition presents with infertility without sexual abnormality. So our answer, that is option A, that is infertility without sexual abnormality is the best described for germ cell aplasia. There are normal levels of LH and testosterone and low levels of inhibin B and elevated levels of FSH and azoospermia. So the answer is option A. Next one. The phenomenon of converting differentiated cells into another cell type by activation of a different set of transcription factors is known as, it is called as transdifferentiation. And the answer is option B. Then which one of the following methods describe probing proteins that have been separated by gel electrophoresis with radio-labeled bait protein and then detecting the interacting proteins by autoradiography? So it is called as far western blotting. That is, answer is option C. Now what is this? It is a method for characterizing protein-protein interaction. So here the method, that is probing proteins that have been separated by gel electrophoresis with radio-labeled bait proteins and then detecting the interacting protein by autoradiography, that is far western blotting. Then western blotting, our usual western blotting uses antibody probe to detect a protein of interest, whereas far western blot uses non-antibody probe which can bind to the protein of interest. So in the question they have not mentioned anything about antibody probe being used. They have told that the following method describe probing proteins that have been separated by gel electrophoresis. So the proteins are separated by gel electrophoresis and then they are treated with radio-labeled bait proteins and then detecting the interacting proteins by autoradiography. So they have not mentioned any antibody here. So it is far western blotting. Now one more thing is, in western blotting, gel electrophoresis usually performed under denaturation condition, that is by using SDS. While in far western blotting, native page, that is without SDS, is used. Then another point is, western blotting is used for detection of certain proteins, whereas far western blotting, it is employed to detect protein-protein interaction. So based on this question, the answer is option C, that is far western blotting. Then Addison's disease is an organ-specific autoimmune disorder in which antibodies are produced against adrenal gland. Answer is option A. Then which of the following statement is true? Answer is option B. The longer the fatty acid chain, the fluidity of the membrane decreases. This is because longer fatty acids are more rigid. So the answer is option B. Then during gastrulation in synapses, the future mesoderm and endoderm move inside the embryo through the blastopore. In contrast, in chickens, gastrulation involves cell moving inward through the primitive streak. Answer is option D. Then match the following. Transduction. Transduction is introducing DNA into bacteria by a virus. Transformation is uptake of DNA into bacteria, and transfection is introduction of DNA into mammalian cell, and transvection is interaction between homologous chromosome segments. So the answer is option A, that is 1, 4, 2, 3, 3, 5 and 4, 1. Which of the following zone has abundant phytoplanktons? So it is the limnetic zone that is having abundant phytoplanktons, because phytoplanktons are it has abundant phytoplanktons. Yes, limnetic zone, because there is it is a zone more heavily responsible for oxygen production within the aquatic ecosystem. So the answer is option C, that is limnetic zone. Then in the equation Kw 55.5 molar and K equilibrium is equal to concentration of H plus into concentration of OH minus. So Kw indicates ionic product of water. Answer is option C. Which one of the following statement hold true for human haploid sperms produced from a diploid spermatogonial cell during spermatogenesis? So all sperms have identical DNA sequences. Need not be. Every sperm has unique DNA sequence. Yes. All sperms have identical SNPs. No. Every sperm has got unique SNPs. Answer is 2 and 4 are correct. SNP is single nucleotide polymorphism. So answer is option B, that is 2 and 4 are correct. Then xeroderma pigmentosum is a rare autosomal inherited skin and neurodegenerative disease mainly caused due to defects in. It is due to defects in nucleotide excision repair system. Answer is option B. Then an individual possessing 2 or more genetically different cell lines derived from a single zygote is known as. It is called as a mosaic. Now they have told it is a single zygote, so it is a mosaic. If it is a combination of 2 different zygotes, then it is called as a chimera. So the answer is option C. Then a bar body is XIST RNA, that is X inactive specific transcript. It triggers the transformation of an active X chromosome into a condensed inactive bar body. Then decondensed inactive X chromosome. It is XIST RNA. Then it is not decondensed inactive X chromosome. Okay, it is an active transformation of an active X chromosome into a condensed inactive bar body. So it is condensed, not condensed. Then polycomb proteins, they are involved in bar body formation. So that is also correct. Then associated with the nuclear periphery. So that is also correct because bar bodies are most commonly situated in the periphery of the nucleus. So 2, 4 and 5 are correct. Answer is option B. Then a 1000 base pair linear and circular DNA has 2 EcoR1 restriction enzyme sites. After digestion with EcoR1 and followed by ligation with T4 DNA ligase, how many combination of DNA ligation products are possible respectively? So when they have told it is a linear and circular DNA. So if it is linear, if you cut with EcoR1 site, it has got 2 sites, so it will give rise to 3 fragments. And the other one, it is a circular DNA, so it will give rise to 2 fragments. Now you join this linear and the circular DNA, you will get different combinations. How? Sometimes the strand will be in the joined with the sense orientation. So from there itself, if you take this, if you consider this linear 1000 base pair linear DNA as your gene of interest, and if you consider the circular DNA as your plasmid vector, then you can see that this particular linear DNA, it will have 3 fragments and it can join with the circular DNA which is cut with the restriction enzyme EcoR1. So it is cut into 2 different fragments. It can be joined to in the sense orientation or even in the antisense orientation. So totally you will get 6, 6 combinations are possible. So answer is option B. Then select a non-denitrifying bacterium. So it is Bacillus ramosus and Thiobacillus denitrificans. They are non-denitrifying bacterium. Then ADF cofflin increases actin turnover by regulating the actin filaments serving. Answer is option D. Then oleic acid contain 18 carbons. Answer is option D. The following method is used to analyze protein post-translational modification of carbohydrate epitope. So it is Eastern blotting. It is a biochemical technique used to analyze protein post-translational modification including addition of lipids, phosphate and glycoconjugates. Then which of the following disease is endemic in India? It is typhoid. Answer is option C. So that's it. Please like, share and subscribe, and thank you for watching my channel.